that screen. There we go. Okay, so we are starting the heart. What is it? It's a big old muscle, but what does it do for a living? It's a pump. A pump for what? To pump blood, that's the chapter we just finished, through where? The circulatory system, or cardiovascular system, which includes the cardio part, which is the heart, and the vascular part, which is what? The arteries and veins, all those tubes that I'm pushing blood through. Remember, blood was important as a transport system. Transport what? Waste, respiratory gases, those big bags of hemoglobin, the red blood cells, not just oxygen, but what else? Carbon dioxide and hormones, antibodies, proteins, nutrients. Yes? Thank you. So it's a transport system, and it transports through the cardiovascular system. So arteries and veins. And how do I hand off the goodies? My delicious cheeseburger or my... through small, tiny little tubes. Tiny little tubes. This is a continuous system. There's no open ends. Yes? We have a pump with a bunch of tubes that leave the heart and then feed back into the heart. And in between is going to be the handoff. So our big tubes will get smaller and smaller and smaller, and then we'll talk about a bunch of little tubes where the handoff happens, and that's the what? Capillary beds. And then they're going to start getting bigger again so they can dump back into the pump. The pump is basically there to push things along. So, where do I find the pump in the cardiovascular system? What cavity do I find it in? It's in the upper region, thoracic, but it's got its own little special area in the middle, in the mediastinum, below the sternum. Yes? Make sure I didn't shut my microphone off. That bone that helps you protect those really vital organs like the heart. And who else is hanging out beside the heart? The lungs. So who protects? Sternum and ribs, that thoracic cavity. So in the mediastinum we see the heart. And it's kind of tilted to the left a little bit. If I have a peak, what kind of section is this? That's a cross section. Well, transverse, yes. We can see up into the heart and its chambers. So it sits kind of snuggled between the lungs, protected by the sternum and the ribs in the thoracic cavity. And basically it's a pump. It's a 4-chambered pump. And we can cut the pump in half. 2 chambers where all the blood comes back to the heart. And again, we're cutting it in half, so there's one on the right and there's one on the left. I'm sorry for you. It's one on the left and one on the right. Yes? We can cut the heart in half as well. The right side of the heart is going to receive blood that's already gone to the whole body, come back to the heart for another push. But what do I have to do after I've gone to the whole body and I come back for a push? I got to dump off some waste products. In particular, I have to dump off some waste respiratory gases. Carbon dioxide. So blood's going to circulate throughout all the tubes, bless you, come back to the heart because it's going to start to slow down a bit, needs another push. But it also had to drop off some waste, respiratory gases, carbon dioxide. So deoxygenated blood is going to come back to the heart. And every time blood comes back to the heart, no matter which side we're talking about, it's going to come back to the top chambers. Those top chambers are called atria. or atrium. Those are the receiving chambers. Then blood has to get pushed out of the heart through another set of tubes. So what are the chambers called that push blood out of the heart? Ventricles. Okay? So the heart itself is a pump located in the mediastinum, covered by a bunch of very wonderful protective membranes in the pericardial cavity, or pericardium. Now what's pericardium? Peri means around, right? So the membranes that surround the heart are serous membranes. And what do they do for a living? They don't produce mucus, they produce fluid, right? And what's the fluid for? Heartbeats, yes? Gets smaller and bigger and smaller and bigger and squeezes up against the lungs and rubs up against them. Yes, rubs up against them. So if I can't decrease the friction by those tissues rubbing against each other, they're going to start damaging the cells. So the layers of the surrounding portions, the pericardium, also produce fluids in spaces to help reduce friction. So the one that goes around the outside of the heart has two layers, the fibrous pericardium, and that's going to be more what? Strong, that's on the outside. Then we have another little layer that is closer to the heart called the what? The parietal layer of the serous pericardium. What's serous? Fluid, right? In this space or pericardial cavity around the heart, what's it filled with? What kind of fluid? What do we call it? It's serous fluid, but we call it pericardial fluid surrounding the heart in that space, yes? Then we have a nice visceral layer that's much softer and more gentle that sits right on top of the organ itself. What's that layer called? It's called the epicardium, or the visceral layer of the pericardium, yes? Remember when we talked about membranes way back when, chapter 4? Yes? Yeah, you remember. What's the difference between what's the difference between visceral and parietal? Parietal lines the cavity. Visceral on top of the organ. Okay, and we call it pericardium because it happens to be around the what? Heart. Yes? The layer underneath is the muscle, myo. The heart muscle is called what? Myocardium. Yes? So we got cardium, and we've heard all these terms before, haven't we? We're just throwing different endings and beginnings on them, depending on where we're talking about. Yes? Epinurium. Ring a bell? Epimysium. Sound familiar? Yes? Same thing. Except now we're talking about the heart. What's the inner lining of the heart called? The endocardium. The endocardium is very, very slick and slippery. How come? Watch this. I'm going to be coming into the heart very fast. Yes? If that's not slick and slippery so I can come in and slide off, if it's kind of rough, what's going to happen when I come in? Yes? I'm going to explode. So, that layer has to be very slick and that hurt. Don't know my own strength. So the endocardium is very slippery and slick. It's one of the things that they had a very hard time replicating synthetically when they were trying to produce an artificial heart. They couldn't replicate in the lab that slippery, slick surface. So when the cells were coming into the artificial heart, what was happening to them? they were exploding, they were bursting because they weren't able to come in and slide in. Okay? And then we have the heart chamber. So you have to know the different layers of the heart. The myocardium, or heart muscle, is in many, many different directions. Because remember, when we studied muscles, a cell does what? Contracts and relaxes, yes? Am I going to be able to make this motion with something that just goes like this? No. So when we look at the cardiac muscle, it's in many, many different directions in bundles around the chambers of the heart, those 4 chambers that we just talked about earlier. So we have the epicardium, the myocardium, the pericardium, the visceral pericardium, and the parietal pericardium. Know what the fluid is and what its job is. And this is pericardium removed, and not next week in lab, but the week after, we'll be dissecting some hearts. Whose hearts are we going to pick on this week? Next week? Week after? Sheep. How big is your heart? About the size of your fist. Yes? So bigger fist, bigger heart. Huh? Yeah. So that's about the size of your heart. And we'll be looking at these in lab so we can get a better view of what these chambers look like. I'll tell you right now, they don't look like anything like these diagrams in your book. That's why we dissect stuff in lab. Because in the book, it makes you think that those atria are these big, huge things, big and round, and that the ventricles are the same size. Is that the case? No, not at all. So, the next section of the book starts to talk about the different chambers and the vessels that leave these chambers. So the atria, and this is the inside of the heart, really cool. See how slick this looks? So the atria are what we call the receiving chambers, and they are on the top of the heart. So blood is going to go around in a big circle and always come back to the pump for a push. Circulate throughout the body, but always come back to the heart for a push eventually. And no matter where I'm coming from, whether it be from the entire body, because I've already made a circuit, I've already gone down to the toes, or I've gone up to the head, or I've gone to the arms and dumped off my load and picked up some waste products on the way, I always have to come back to the heart for a push. Otherwise, I'm just not going to be fast enough to get through the body if I don't get a push from the heart. Yes? All right. So wherever I've been, I've gone, I've done my thing, I'm coming back. So I have 2 large vessels, and every vessel that brings blood back to the heart, no matter where it's been, is always called a what? A vein. Do veins always carry deoxygenated blood? No. So don't remember it that way, because there's exceptions to that rule, and we'll talk about that in a minute. But any big blood vessel that comes back to the heart and dumps into an atria, whether it be on the right side or the left side, is always going to be called a vein. So dumping from the bottom section, remember my heart's here, right? Draining all the stuff that's already traveled through the bottom section is called the inferior vena cava, and everything that's dumping from above the heart, superior vena cava. And those are going to dump into the right side of the heart. And the right atria is where veins are always going to dump into that have already gone through all the circulation, already been through the body. Why do you think I'm going to try to separate this pump somehow? Yeah, some comes in, some goes out. It's kind of going at the same time all the time. Blood is coming into both sides, both atria at the same time, because I'm kind of flowing through a circle. But why don't I want the right side and the left side to mix? Yeah, when we're talking about those respiratory gases. So what I want you to remember is this pump, this 4-chambered pump, is actually a 2-sided pump. So we're going to cut it in half. The right side is going to be responsible for receiving blood that's already gone through the system, and then taking that blood and pushing it where? I got to get rid of the waste gases. Where am I going to do that? In the lungs. So the right side of the heart we call the pulmonary pump. Yes? All right, so I get all the blood from the whole body into my receiving chamber, atria, right atria. I'm also going to have little valves that separate all of my chambers as well. Because what don't I want to happen? I don't want blood to go in the opposite direction. I want it to keep moving forward. So these little valves between the 4 chambers and leaving the heart are going to prevent backflow. The one on the right side has 3 little sections. Oops. Well, you know what they're called? Or what that valve is called? It's called the tricuspid valve. It's called the tricuspid valve because it's got 3 flaps. And the 3 flaps look like this. You see that? These are little what? Cusps, yes? Or flaps. So it's called the tricuspid valve because it has 3 flaps. And that is also called an atrioventricular valve. Why do you think we call it an atrioventricular valve? Because it's between a atria and a ventricle. Very good. So when blood then travels in here, this atria will eventually contract and push its volume into the next portion of the heart, which is the ventricle. This is the right ventricle. Notice it's separated from the left ventricle. Yes? That's called the interventricular wall or septum. All right, so blood's then going to come through into the right ventricle. Now, when I'm in a ventricle, where do I go next? I go out. So in your book, it talks about the discharging chambers, the ventricles. When the heart or ventricles contract, and remember, they're both contracting at the same time. So what's going to happen during a one cardiac cycle or heartbeat? Atria are both going to contract and relax, and ventricles are both going to contract and relax. Yes? We'll talk about cardiac cycle in a minute. So once I've traveled to the right, what is this again? Ventricle, and the ventricles contract, I am then going to be pushed out of the heart. Any big vessels that leave the heart are called what? Arteries. But this right side of the heart, what did we call it again? The pulmonary pump, is going to send blood where? To the lungs. So it's going to send, and that's why this is blue, because what kind of blood do we have here? Deoxygenated, but that's not a vein. What is it? It's an artery. There's a valve here to prevent backflow when these guys relax. These valves in the arteries that leave the heart are called semilunar valves. You know why they're called semilunar valves? They look like little half moons. Exactly. The one that separates the right ventricle from the pulmonary what? Artery is the pulmonary semilunar valve. And we'll talk about the other guy in a minute. So we're going to send blood now to the lungs. And what's going to happen in the lungs? Now, there's the heart, there's the lungs. Do we need a huge push? No. So the wall of the right ventricle is much thinner. I want to point that out before we get to that side. So I go to the lungs, I do my thing. What am I doing in the lungs? Dropping off carbon dioxide so I can breathe it out. Picking up oxygen. Yes? Who's going to help me do that? My little bags of what? My little bags of hemoglobin. My who? My erythrocytes, my red blood cells. So everybody's going to get off the ride, those little carbon dioxides, get breathed out. And everybody, oxygen that I breathed in is going to get on the ride and then take a trip back for a push. So I'm going to go back to the heart for a really big push. Because where do I have to go now? All over the place. Toes, fingers, head. Yes? So now that wimpy little push I got from the right side isn't going to do it. So I got to come back to the heart, and I'm always going to come back to the heart into a what? Receiving chamber through a vein. Which veins? Pulmonary veins. And now, now they're red, which indicates oxygenated blood, yes? They're coming in from both sides. Remember, I have two sides, yes? So see my pulmonary vein? It branches off to both sides, two sets of lungs, yes? Comes back to the heart, dumps into always a receiving chamber, atria, except this time I'm going to be where? On the left side. Again, I have a valve that separates the two, but this guy is not a tricuspid valve. This guy is looking like this. Bicuspid valve. Yes? It's also an atrioventricular valve, and it also has another name. It's also called the mitral valve. Ooh, that would be a good bonus question. What are the three names for the right valve between the, or the left valve, excuse me, between the atria and the ventricles? So I can be called an atrioventricular valve, or I can be described by my cusps, either a tricuspid on the right or a bicuspid on the left, or the left one also has another name, mitral valve. So I get into the atria contract and push me down into where? The left ventricle. The left ventricle, and again, this is why we do dissections, is most of the heart. So the right one, realistically, is like this big, and the left one is this big, and has much thicker muscle. How come? I need a huge push now, because now I have to get blood from here to there. And I have to have enough pressure to get me back. Yes? See why we need the heart? All right. No, actually, we're going to have some other help on the up part that we'll talk about. We just got to get it out and moving. So, the left ventricle is most of the heart. So when we actually look at the heart in lab, we're going to see it's mostly left ventricle. The walls of the atria are much thinner. They don't have to absorb as much pressure, and they don't need to give as much of a push. When we push out of the left ventricle, we're going to push through another valve. What's that called? It's the aortic valve, or the aortic semilunar valve, those half moons again. And the big artery that leaves the heart, and this is going to be really thick to absorb all the pressure that we just got from the push from the left ventricle, is called the aorta. And what's going to happen to the aorta is we're going to start to see branches off of it. So there's branches that go up, and then a big section that dips down. That's called the descending aorta. And there's going to be a whole bunch of branches off of that. All the branches are so that everybody in the body can get what? all the stuff that's needed in the transport of blood. So oxygen, get rid of my wastes, get nutrients, get rid of wastes, and so forth. Yes? The order in which I just said all of this movement, you will need to repeat. So, you need to know how blood travels through the heart and the pathway of blood through the heart. Oh, by the way, see in real life what we see? So the atria, or auricle, which is part of the atria, very small, thin-walled. The right ventricle, much smaller than the left, which is most of the heart. And then the interventricular septum, which separates the two. There's also a wall that separates the two atria. What do you think we call that? The interatrial septa. So this little diagram on page 668 in your textbook gives us kind of the direction in which we go. And I'm always going to start with everybody comes back to the heart. That's always going to be the beginning. So I'll go quickly over that. Listen to it again. You start by coming back from the system. So what do we call this side of the pump, by the way, before I lose train of thought? This is called the systemic pump. The left side goes to the system. The right side goes to the lungs. Pulmonary pump, systemic pump. Yes? All right, we're always going to come back to the heart through the atria. So, inferior, superior, vena cava, dump into the right. And you always have to make sure you get the right and left in your description. The right atria. Then I go through the tricuspid valve into the right ventricle. Then I go through the pulmonary semilunar valve, out the pulmonary what? Arteries to where? The lungs. I'm going to do my thing there. I'm going to come back to the heart through the pulmonary veins, dump into the left atria, through the bicuspid valve or mitral valve, into the left ventricle. Did I say right or left? I said left. Okay. Left ventricle. Then I'm going to push out of the heart through the aortic semilunar valve, out the aorta to the system. Yes? Will you see that on your test? Absolutely. So most of the heart, when we look at the heart, is mostly left ventricle. Very, very thick myocardium. What's myocardium again? Heart muscle. And that's what needs a lot of oxygen. So we actually have to make sure that we give dibs to this muscle first on the oxygen before our travels. Even before the brain. And only because it's physically that way. The brain is actually going to get quite a bit of blood volume, and it's going to get it pretty quickly. Yes? See where we are? Kind of built that way for a reason. So when blood leaves the heart, remember that big, thick aorta? Those branches that go up are going to really quickly come here. Because remember, we're getting one hell of a push down in the ventricles, right? So it's going to hit the head fairly quickly. And we'll talk about some of the major blood vessels that are going to bring blood to the head very quickly. So about 20% of your blood volume goes there quick. So most of that muscle, myocardium, we need to make sure it gets plenty of oxygen, yes? So we have special blood vessels that are going to serve it very quickly. Coronary what and coronary who? Coronary arteries are going to take blood away from the heart and bring blood where? To the heart muscle itself. So we see the left coronary artery coming right off of the aorta, and that's it's kind of tough to see that. It looks like it's coming off the pulmonary trunk, but it's not. Coming off the aorta, behind. Okay? And the aorta here. So we see the right coronary artery and the left coronary artery that then branches to feed the heart. And what happens after we've dumped off our goodies in the heart and picked up waste products from the heart? Cardiac veins. And where are they going to dump into? They're going to eventually dump back into the vena cavas through this coronary sinus region. Okay? Now when we talk about blockages in these arteries, what do we have to do to fix that? If we get a blockage in them because, oh, maybe cholesterol gets dumped off there and blocks the flow, and that's not good because then the cardiac muscle itself won't get the food that it needs. We have to do something called bypass. So we have to kind of bypass the blockage with some clean tubes. Does anybody ever know of anybody who's had coronary bypass surgery? And they talk about how many bypasses they need, like a triple bypass or a quadruple bypass. Have you ever heard terms like that in TV shows? Okay, so they're just saying how many different places had blockages and how many different bypasses they had to do. They actually go in and they get some nice vessels, usually from the leg region, and make a little bridge so that we can bypass the blockage, cut that out, and make sure everybody gets the goodies they need in the heart. If the blockage prevents blood from going to a certain section of the heart, what's going to happen to those cells? They're going to die. What's that called? A heart attack or a myocardial infarction. Okay? And that's because the cells are starting to die. That pump is not going to work as efficiently when those cells or massive cells start to be destroyed because they're not getting what they need. They can't get rid of their wastes and they can't get the nutrients they need. Can that be life-threatening? Yes, it can. And coronary artery disease is high up there on the death toll for people in the United States. More people die from coronary artery disease or cardiovascular issues than they do of cancer. Yeah. McDonald's and Burger King. All right, so coronary circulation then, described in your book, is extremely important. You will have to do some identification of these vessels, and that will happen in your lab practical of next. Yes? So they talk about blockages, heart attack, infarction, under homeostatic imbalances of the cardiac circulation. The atrioventricular valves are described next, and again we can see them nice and clearly. The tricuspid valve has what? 3 little flaps. We see that here. And the bicuspid valve has 2 flaps. Again, those are atrioventricular valves, valves between the atria and the ventricles. And one of the things that are associated with these valves, and these are up close and personal real ones, is these little strings that actually help the valves open when it's time for them to open, and they release like a little trap door to help them close when back pressure starts to build up. Those little strings, or heartstrings, there was actually a song way back when in the old days about the heartstrings. And that's like even way before my time, so. These strings are called the chordae tendineae. And they are going to help open and close those valves when need be. When do I have to close the valves? The blood's gone through, but who starts to contract to push the blood out? The ventricles. If I don't close the door, what's going to happen when the ventricles contract? Everybody's going to flow back into the atria. Do I want that to happen? No. I want everybody to leave through the semilunar valves, right? And out those arteries. So that's when the door shuts. the atrioventricular valves. The semilunar valves are going to do the same thing for us, but they are going to be found where? One is going to be found leaving the left ventricle, and between the left ventricle and the big artery that leaves the left ventricle, that's called the what? The aorta. So aortic valve or aortic semilunar valve. And then we also have one that's leaving the right ventricle, going to the lungs. So it's between the pulmonary arteries and the right ventricle. It's called the pulmonary valve or the pulmonary semilunar. Not the tricuspid, those are atrioventricular valves. The pulmonary semilunar valve. Yes? Everybody with me?
Thank you.
You're welcome. Oh, she's got a runny nose. All right, so tricuspid, mitral valve or bicuspid. Do you see the chordae tendineae there? Little heartstrings. They're attached to these little muscles down in the ventricles called papillary muscles. So when they contract, they pull on the strings, and what happens? The trapdoor opens. When they relax, ping, the string lets go, the trapdoor shuts. So they're going to help keep blood flowing in the right direction. Oh, let's have a little clicker quiz. Yeah, I'm being for real. JK. I know what that means. All right, of the following layers of the heart wall, which consumes the most energy? Oh, hurry up. Yeah, we just talked about it. Yeah. Uses the most energy. What do you get? Take a guess. It would help if you paid attention in lecture, see? Now I'm going to make sure you pay attention in lecture. It's my new and improved clicker question quizzes. That my number 36? Remember, this is also your attendance. So click, or you're not here. If you forgot your clicker, tough toenails. You're not here either. Yep. So 39 is my number. 5, 4, 3. Click the button. 2. 40 is my number. Everybody's clicked. Who can click? All right, what's the answer? B. Yes, B. Yay! B is the answer. So the muscle, the myocardium, is what's going to use most of the energy, or make most of the energy, and consume most of the energy, right? Because it's doing all the what? It's doing all the work. Huh, sneaky, huh? All right, so that's it for now. So atrioventricular valves, semilunar valves, you should know where they are, and you should know the difference between them. The next thing the book talks about is the atrioventricular. Yeah, this one is just showing you how blood flows through the heart and how those valves are going to act. So blood returns from the heart, atria fills, blah, blah, blah. And then we see the semilunar valves. So this diagram is showing us the difference between the atrioventricular valves, where they are in the heart, how they open and they close, and then the semilunar valves. Oh, another question! Which of the following structures is an exception to the general principle surrounding blood vessel oxygenation levels? So typically people say, oh, all the arteries have oxygenated blood and all the veins have deoxygenated blood. Nuh-uh. So this question is saying, what are the exceptions or exception to that rule? 5, 4, 3, 2, 1. Just click something. Come on, number 40. All right, too late. What's the answer? D. The answer is D. So remember, the exception, you know what, if you click, you can click again. The last thing you click is what's going to register before I stop. So if you clicked and then you said, oh crap, that's not the right answer, and click another one, that's the one that's going to register. Okay. Well, sometimes. So remember, arteries don't always carry oxygenated blood because the pulmonary arteries carry what? Deoxygenated blood to where? To the lungs. And the pulmonary veins carry oxygenated blood back to the heart. So the rule is a vein always carries blood where? Back to the heart. The artery always carries blood away from the heart. Yes? So the next thing that the book talks about are muscle cells. Now we talked about cardiac muscle when we talked about tissues way back in chapter 4. How did we describe cardiac muscle? It was striated, and everybody knows why it's striated. Why? Yeah, you do have to remember this stuff. Because of the way the sarcomeres are arranged in the myofibrils. Yes, that's what you were going to say, right? Absolutely. Yes? So remember the I band and the A band and the Z line and the H zone and all of that stuff that lined up all nice and pretty in the myofibrils, the little tiny fibers inside the muscle cells? Yeah, that's what causes the stripes. So they are striated. What else do we use to describe them? Branching! So if they're not straight across like telephone poles, like skeletal muscle is, why do you think they branch? I need to squeeze like this. I don't need to go, right? So I need to go in different directions to create that motion, and that's what creates all the different layers and directions of muscle cells in the myocardium. We also said they were singular nucleus, not multinucleated like the skeletal muscle cell was. Sometimes we'll find one with 2 nuclei. They're also, are they voluntary or involuntary? Involuntary. Who controls their contraction and relaxation? The autonomic nervous system. As a matter of fact, they need to contract and relax 24/7, don't they? Yeah, it's kind of important. So they have their own little group of cells that are going to help continue that contraction and relaxation. They're called the autorhythmic cells. And we're going to talk about them in a minute. When they hook up together, they're going to make a real tight hookup. Yes? They're not just going to sit like this. They're going to hook up really tight. Why do you think they need to hook up really tight? Exactly. They're constantly contracting and relaxing and pulling on each other. So when they hook up, they're going to create a really thick hookup called the intercalated discs. Does that ring a bell? Okay, when we look at them under the microscope, if you look over here, you can actually see where they're joined. They create these little stripes, big thick ones, up and down. Those are called intercalated discs. And what we're going to find inside there, those intercalated discs, why don't we have an up close and personal? Oh yeah, they're there. We find little straws that join the two cells that are joining up. That kind of join their cytoplasm together. Remember that cellular junction called a gap junction? Oh yeah, we had to remember that stuff. We're going to find a lot of gap junctions in there, as you can see in that diagram. And then we're going to find a lot of really strong holds or spot welds. What do we call those? Desmosomes. So that's why we see them under the microscope thick, because there's a lot of stuff going on there in that connection between two cardiac muscle cells. So, what's going on in there? Well, there's a whole bunch of chemistry going on. Depolarization, repolarization, with the help of sodium channels and calcium channels. And everybody remembers what's involved in a muscle contraction, right? Yeah. You remember when we had some sort of impulse that was going to open the sodium channels, and sodium was going to rush in, causing a paralyzed pump in the terminal cisternae so that calcium could rush out and come in contact with the regulatory protein, troponin, which was going to pull tropomyosin away from the myosin binding sites on the actin molecules. Myosin would grab on to the actin molecules and undergo a power stroke. Then ATP would attach to that myosin head, releasing its grip, and undergoing hydrolysis would release the energy and put it down in its high energy configuration. Remember that? Yeah, you got to remember that again, because that's what's going on here. Except it's going on a little bit differently, because muscle cells, cardiac muscle cells, are constantly contracting and relaxing. So with the help of some slower channels, the chemistry is going to even out itself a little bit better. And what we're going to see is a much longer refractory period. Everybody remembers these, don't you? Remember the whole zap? Go from -80 to hit threshold, -55 millivolts. Once I hit threshold, I was going to cause the whole depolarization thing to take place. Yes? Yeah. Okay, same thing here with this cardiac muscle cell. Except what we're going to see is we're going to see a much longer refractory period or plateau. That's what allows these cells to constantly contract and relax. Okay? So the chemistry happens and gets back and can happen again because of that plateau. So we see it a little bit different to tracing when we look at the chemistry that's going on in a cardiac muscle cell versus a regular old skeletal muscle cell. When you bought your book, how much did you pay for it? Lots and lots of money, yes? Well, one of the things that you purchased when you bought this book was something called Interactive Physiology. Crack it open for this chapter. It's going to be one of your good friends. So go look at the Interactive Physiology module, Cardiovascular System. Yes? So, in order to constantly give us a nice even contract and relax and contract and relax and give us a nice plateau between, we have a group of cells that are going to help keep the heart beat on a regular basis. And this is what we're going to talk about when we talk about things under heart physiology. In particular, I want to make special or give special thanks to your conduction system. On page 677, figure 18.14, we see a description of the cardiac intrinsic conduction system, an action potential succession during one heartbeat. So basically what has to happen during a heartbeat? is I have to contract the atria to push out the volume, and they're going to do it at the same time, right and left side. I'm going to then push that volume with the contraction down into the ventricles. These guys have to then relax for the next group of cells that comes along. And what has to happen next? I have to contract the ventricles so I can take the blood that squirt out, or down, I should say, from the atria into the ventricles, and I have to push it out of the heart. To do that, the ventricles then have to contract. This, and those series of events, we're going to talk about in a moment when we talk about the cardiac cycle. But the group of cells, with the help of the autonomic nervous system, that's going to help keep contraction and relaxation at a nice normal pace, is a group of cells called the autorhythmic cells. Autorhythmic. What's it mean? Automatic rhythm, exactly. So these cells are going to ensure that we continue to contract and relax these muscles at a nice rhythmic rate so that we can have blood move through the heart efficiently. The first group of cells is what we call the Sinoatrial Node. Again, these cells are autorhythmic cells, and they're going to basically cause a chain reaction of chemistry that's going to set off muscle contraction. And I already just described muscle contraction to you, right? Yes? If you don't remember, you need to refresh. So the sinoatrial node is going to be the beginning of the impulse that's going to cause the atria in a wave to contract. That message is then going to get sent down to another group of cells that are going to then continue the wave of impulse. And that is going to be the Atrioventricular Node. So it's a group of cells between what? Between the atria and the ventricles. Now remember, my ventricles are really big and I have a lot of space to cover. So the Atrioventricular Node is then going to pass that wave of contraction on to an area called the atrioventricular bundle. And that's going to be found in the interventricular septum. Yes? What happens to the atrioventricular bundle? Who does it pass its message down to next? The bundle branches. Now, I have two ventricles where I have to cover a lot of space. So the bundle branches are going to branch off into left and right bundle branches. And then they're going to send their message to the groups of cells that are going to hit the ventricles. What do we call those guys? They're Purkinje fibers. So that depolarization of the heart muscle will happen in a wave, first the atria and then the ventricles. Yes? And these cells are going to help ensure that that happens in a nice, smooth, even way. So should you know the pathway of that intrinsic conduction system? Yes. Sinoatrial node is going to kind of start to get the ball rolling. So another term used to describe the sinoatrial node is the pacemaker. What's a pacemaker do? If you have trouble with your conduction system for whatever reason, there could be a left or right bundle branch block. Yes? What do you need to do? Because if your heart doesn't contract properly, you're kind of toast. Yes? You might have to go and have a pacemaker put in. And what is it? It's going to start with a little zap, and you got to go get your batteries changed every once in a while, yes? With a little electrical impulse to get all of these pathways going. Sometimes the rhythm is off, and if the rhythm is off, the pumping action is going to be off as well. So know the intrinsic conduction system described on page 676, 677. Know the order. Who's going to keep the pacemaker in check? The central nervous system. So the vagus nerve cranial nerve what? 10. Woo! Can help the parasympathetic division of the autonomic nervous system. And what was the parasympathetic division for? Slowing things down. And what's the sympathetic division for? Speeding things up. So when I find out that there are hermits being sold in the bookstore, who's going to kick in? Sympathetic. Sympathetic cardiac nerve's going to do what to my heart rate? How come? I need more oxygen for my muscles so I can run to the bookstore and get my hermits. Yes? Once I have purchased my hermits, I can relax. Who's going to kick in? Do I need to be going that fast anymore? No. Now my digestive system needs more blood so the heart can slow down. Yes? Yep. All right. All of this change is chemical change. Electrical events are happening. Electricity. You know, when we talked about nervous impulses, nervous system, we're talking about electrical events. Yes? You can also pick those electrical events of those autorhythmic cells up on something called an EKG, or electrocardiogram. So on page 679, electrocardiography describes the impulses that we can pick up on an electrocardiogram, or EKG. We call it EKG. It's actually ECG, but you know why there's a K there? Did you ever try to read a doctor's writing? I'm not kidding. That's why we do this. It's really hard to tell the difference between a C and an E, because there's another test, an EEG, so that's why the K's there. But it's the same thing, electrocardiogram. I don't know if they actually did that, but yeah, it's a different language, but that's what I like to say, because you can't read doctor's writing. So that's why the K's there. But anyway, you need to know what's happening during what we're picking up here in these impulses. So remember, during a cardiac cycle, which we're going to talk about in a minute, there's a couple of different events that are going on. The atria is contracting, the ventricles are contracting, and then they both have to relax at one point during a cardiac cycle. This is one cardiac cycle. From the beginning of the P wave, this guy here, to the beginning of the next P wave, that's one full heartbeat. We can't see the next P wave here, but that would be one cardiac cycle, one heartbeat. So what are we picking up here? Well, the first bit of change, chemical change, electrical events that we can pick up on a cardiogram is the beginning of the cardiac cycle, which involves atrial depolarization. What the heck does that mean? The atria are doing what? Contracting. Depolarization. So that's your P wave. What has to happen next? Well, two things are kind of happening next. Remember that intrinsic conduction system? I'm sending message down that atrioventricular bundle and the bundle branches and the Purkinje fibers to cause what to happen? The ventricles to contract. Much bigger wave, yes? QRS. But I also am doing something else that's kind of hidden in there. We can also pick up the relaxation or repolarization of muscle. So while the ventricles are contracting or depolarizing, what are the atria doing? Repolarizing. So we should also put in here what? I hate this thing. Atrial, what? Repolarization. No, it takes place within it. I just put it there because that's where I could fit it. Okay? So that's why it's so big. QRS. Yes? What happens next? So the atria contracted. The atria relaxed, ventricles contracted. Now what has to happen? Ventricles have to relax. Ventricular repolarization is picked up in the what? T wave. P, Q, R, S, T. Shows us a cardiac cycle. And the PQ interval and the QT interval and the ST interval should be in a certain amount of time to have efficient movement of blood through the heart. Question?
Yeah. This would be like an extreme normal.
Correct. Now when you look at a cardiogram, and there's some pictures of them over on page 681 in your textbook, we actually make it on this very specific graph paper. And when a cardiologist is reading a cardiogram, they have a little ruler with them to measure the intervals. What if it takes too long between atrial depolarization and ventricular depolarization? Could there be a problem moving blood through the heart? Yeah. So these time intervals are very important in normal movement of blood through the heart. So they are actually measuring those, and they are described in your book. So we have the QRS complex, the T wave, the PQ interval, the PR interval, the ST segment, and the QT interval. We will ask you questions about these in and on your lab practical exam, for sure. So this is a nice little diagram because it ties in what's happening during these different electrical events that are being picked up on the EKG. So again, atrial depolarization happening during the P. Then we start to send message down the atrioventricular node to the septum, interventricular septum, and the bundle branches. So we start to see where it moves then. Then QRS complex sending message down the bundle branches up around the Purkinje fibers to cause ventricular contraction. But don't forget, what else is happening while the ventricles are contracting? Yeah, the atrial are what? Or the atria are repolarizing. So don't forget that. That's also in the complex. We can't see it separated because there's just too much electricity going on. So we can't see two separate peaks. They're all kind of encompassed in the one. Then we have message being sent and being repolarization taking place. And T wave shows us ventricular repolarization. So one cardiac cycle is from the beginning of the P wave to the beginning of the next P wave. So here are some, and these we can't see the peaks as well because these are what we call rhythm strips. But a normal sinus rhythm would see nice regular P, QRS, and T waves going on at a nice regular time interval. What happens when we start to get weird time intervals? We're going to have problems pushing blood through the body. We can, through a cardiogram, see electrical problems with the heart. The time intervals are going to help us tell a little bit about the integrity of the vessels or the chambers themselves. If I'm way flabby and it takes me a way long time to fill up and then contract, I might have a longer time interval. If I have too quick a time interval, I might have problems pushing blood through the heart. So we can see lots. This guy's in big trouble here, right? Holy moly. Fibrillation, what is that? Instead of shoo, shoo, I'm going ee. Is that good? I can't push much of anything doing that. So if the atria are fibrillating, atrial fibrillation, it's not as bad because who's going to still contract? The ventricles. So all of that pressure will eventually push some of the blood through, but atrial fibrillation can be a problem because it can start to cause a patient to not only not move their blood through their heart efficiently, but spontaneously clot. Yeah. You didn't get my description? But when the ventricles do that, you're toast. How come? There's no volume getting pushed through the tube. So everybody's backing up behind you and nobody's getting pushed out. So ventricular fibrillation, very, very bad. Okay? We can also pick up, again, blockages in the conduction system as well. So the EKG is a real quick way to assess what's going on with a patient's heart muscle. What's happening when they have a murmur? I'm going to tell you in a minute. Now, I don't have a door here. When I listen to the heart, what do I hear? What's lub-dub? Is it the blood moving through the heart? No, you can't hear that. It slips too fast. Aha! It's the valves closing. If I had a door, and I don't have a door, and I'm afraid that I'll make an alarm go off. All right, listen to this. Shh, listen. I gotta get there. Hold on. All right. Now wait a minute. Listen after when I tell you to listen, start listening, okay? Not now. Okay, now listen. Did you hear anything? No. Listen now. Did you hear anything? Door closed. Can you hear the door open? No. When the door closes, you can hear it. Yes? So what am I talking about doors? The valves. We have 4 of them, right? Why are there only 2 sounds? They open at the same time and they close at the same time. So when the atrioventricular valves open and then slam shut, that's the first heart sound, lub. When the semilunar valves open and then shut, that's dub, the second heart sound. Yes? So that's what you hear. You can't hear the blood flow through the heart. But sometimes, if the valves don't close properly, you can kind of hear a little swish of blood backflowing through the valves. And that's what? That's a heart murmur. Sometimes the valves don't close properly, or you might have little holes where they shouldn't be. And you'll hear a swishy in and amongst the doors closing. And that's what they describe under homeostatic imbalances and heart murmurs. So in order to listen to these sounds, and that's what the doctor does when he listens to your heart, he takes a stethoscope and sticks it right on your sternum, right? No, because you can't hear anything through that big bone. So what do you have to do? And that's what this shows us. So when the doctor listens to your heart, and pay attention next time, he puts his she puts his stethoscope in 4 different places. Yes? Listening to what? The valve sounds. Then they're listening, they can listen to heart sounds too, but usually it's what? Lung. Yes? One of my favorite diagrams in the whole textbook. My favorite. You know why? Because in one diagram there's so much information. So at the top, you already know about the electrocardiogram, right? So we talked about the P and the QRS and the T. Right up there in the same portion, we see at what point in the cardiac cycle, and that's what this diagram is. It's called the cardiac cycle. And it's showing you everything that happens during one heartbeat. And where it happens. Right up there, the first heart sound. What was that again? The atrioventricular valves? Closing, yes? First heart sound. Follow it down. Look. What happens to pressure in the atria? They go down. Back pressure from the ventricles, which is starting to go up, causes the doors to Slam shut. Yes? The pressure builds up in the ventricles. What's happening now? After QRS depolarization, pushing that blood volume up, or out, excuse me. What happens next? Yeah, so they relax after they push their volume through what? Semilunar valves. Woo, they're down here too. Yes? So when the pressure starts to release, what's going to happen to the semilunar valves? They're going to shut. Second heart sound. Yes? See what happens to the pressure in the system? See when the atrioventricular valves are open, when they're closed and open again. See when the aortic, which is the semilunar valve that connects the left ventricle to the aorta, and the pulmonary valve, right ventricle to the pulmonary arteries. Yes? You see when they're open and they're closed. These are the phases of the cardiac cycle. They have names. So during the first phase of the cardiac cycle, we see the ventricles filling because the atria are what? Whoosh, squishing all their volume out. So the phases of ventricular filling, mid to late diastole. What's diastole? She's throwing in all these terms. You know when you talk about blood pressure? And then there's a couple of terms they use. Systolic over diastolic. What the heck do they mean? Systolic is the same as saying what? Contraction. Diastolic is the same as saying relaxation. So, ventricular filling happens mid to late diastole of who? If the ventricles were in systole, would they fill up? So when the ventricles are relaxing, now I can accept what? Blood from the atria. Yes? The second phase, we're going to break down into two parts. Isovolumetric contraction phase. And what's happening to the pressure inside the ventricle? It's starting to go up. Contraction. A. B. Ventricular what? Ejection phase. Everybody's leaving. The pressure is going to start to come down. Ventricular systole. Ventricular what? Contraction also involves what? Relaxing. Atrial diastole. Yes? You with me? No. This is one you got to kind of stare at for a while, and then you're going to go, oh, that is my favorite diagram in the book. No, everything. Whole blood volume. Plasma, blood cells, the whole kahuna. Yes? So that's the second phase. Ventricular systole. Also, don't forget, the atria have to relax because what do they have to do next? Now they have to fill up again. So the last phase, early diastole, everybody's kind of relaxing, but stuff's going on. Isovolumetric relaxation involves blood being pushed, everybody being pushed from behind, allowing the ventricles to do what? The ventricles need to be relaxed when they're filling up, don't they? Because if they're contracting, they're doing what? Pushing their volume out. So they're in relaxation, they're filling up, and then we start all over again. Yes? So have I blown your mind yet? Okay, whew, good. That's what I wanted to do. So, what I need you to do this weekend, okay, because really, we should be done with this, but yeah, we got a little bit longer to go, a little bit more to talk about before we hit the next chapter. Are you going to have a quiz on Tuesday? I know, he's giving me the eye. She's evil. That's a yes. Yeah, that's right. Valentine's Day is when? Tuesday. So we're going to have a quiz on chapter 18 on the heart. No, it just worked out perfect, didn't it? On Tuesday, Valentine's Day, we will finish up the last little bits, but you really need to stare at that diagram and understand what's going on during a cardiac cycle. So, we're going to what, hon? What do you want to do? You want to finish the chapter before and then have a quiz? I don't know. We'll see. That's what, if you look under the Purkinje, the bundles should be under there. You know, it's the Purkinje Fibers are, all those little guys down the bottom, it should say bundle of His. And yeah, you're right, it did leave it out. It's in the old book. All right, so my friends, study hard, quiz on